Imagine you're trying to piece together a massive jigsaw puzzle, but you can only look at small sections at a time. That's somewhat analogous to sequencing a whole genome. We can't read the entire DNA sequence in one go. Instead, we get many smaller overlapping fragments that can be arranged to give the entire genomic sequence. Physical mapping is a method used to determine the physical positions of DNA sequences on a chromosome. Physical mapping aims to establish the actual physical distances between genes. Now, let us understand how physical mapping is done. When scientists want to create a physical map, they take the entire genome and break it down into many overlapping fragments. These fragments are then inserted into the BAC, YAC, or cosmid vectors and cloned. The key to forming contigs is identifying where these cloned fragments overlap. This is done by looking for shared markers or overlapping DNA sequences. By identifying these overlaps, scientists can arrange the cloned fragments in their correct order along the chromosome. A contig is formed when multiple overlapping fragments are assembled into a continuous sequence. When arranging the overlapping fragments, some gaps may form in the sequence. These may include sequence gaps, which are regions of the genome for which we don't yet have a contiguous DNA sequence within our physical map. They may be physical gaps, which are parts of the genome not covered by any of the cloned fragments in our physical map. Let's head to the concept of sequence-tagged sites. Imagine our jigsaw puzzle analogy again. We've managed to assemble several smaller sections, contigs, but there are still gaps between them. How do we figure out the order of these larger sections and how far apart they are? That's where STSs come in. STS is a short DNA sequence, typically 200 to 300 base pairs long. This DNA sequence itself is designed to be found only once in the entire genome. The real power of an STS lies in the fact that we know the short DNA sequences that flank it. These flanking sequences are used to design specific PCR primers. When PCR, a polymerase chain reaction, is performed using these primers and a DNA sample containing the STS, only the region between the primers, which includes the STS, will be amplified. Consider we need to map the genes on a chromosome. When STS is applied and overlapping fragments are formed, we get the following labeled fragments. If we consider fragments 1 and 4, they have the same STS marker in their fragments. Fragment 4 has an extra STS marker, B. This means that we can arrange the two genes adjacent to each other. We can see that fragment 2 will be next to fragment 4. This is because the two fragments overlap and the STS markers, B and C, appear next to each other in fragment 8. This means fragment 2 will follow fragment 4 when aligned in the sequence. In a similar manner, we can arrange fragment 5 next to fragment 8, fragment 9 next to fragment 5. The results of the screening are compiled and the overlapping fragments are aligned, allowing the scientists to fill the physical gaps by comparing the STS overlaps and the contigs. If two different contigs are found to contain the same STS, it strongly suggests that these two contigs are likely to be adjacent or overlapping in the actual genome. Why? Because that unique STS sequence can only exist in one place in the genome. If we find it in two separate contigs, those contigs must be derived from a region that spans the location of that STS. Contigs are likely to be adjacent or overlapping in the actual genome. What is the significance of physical mapping? Mapping provides clues about which chromosome contains the gene and precisely where the gene lies on that chromosome. It can offer firm evidence that a disease transmitted from parent to child is linked to one or more genes. The relative distances between positions on a genetic map are calculated using recombination frequencies, but actual distance can't be measured, but physical mapping is able to give precise distances.